The next portion of our agenda is a moderated panel discussion by of NCCN guideline panel experts. And we're really fortunate to have with us today Dr. Clifford Goodman as our moderator. Dr. Goodman is Senior Vice President at the Lewin Group, National Healthcare and Human Services Consulting firm. He has over 30 years of experience working with government, industry, and nonprofit organizations in the US and abroad. So with that, I'll turn the moderation over to Dr. Goodman.
Thank you, Dr. Carlson. Glad to be here. This is our panel on Meet the NCCN Guidelines Panel Experts. As many of you know, perhaps most of you know, the NCCN guidelines have become basically the industry standard in oncology, and certainly for those who care about the healthcare system and how it works, payers tend to look to, public and private payers tend to look to the NCCN guidelines first, insofar as getting that expert input on when and how to reimburse, how to cover interventions in oncology across a broad variety of areas. And we're going to cover several main topics today, and I just want to let folks know up front kind of what we want to cover, and then I'm going to have our panelists introduce themselves. We're going to have a quick introduction of the program by Dr. Carlson, whom you've met, and get a little bit into the guideline processes. How does the guideline process work? Who's involved? How long does it take? How do questions come up? How do you handle evidence? Those sorts of things. We're going to talk a little bit about how, and this kind of plays off the presentation you just heard from Dr. Stewart, how has the COVID pandemic affected the thinking about the guidelines they're updating in their content? We're going to take a good look at real-world evidence and real-world data and to what extent those are incorporated into guideline updates. We want to take a special look at disparities in care. This has obviously been highlighted by the pandemic, but certainly NCCN panels have been looking at how to manage disparities in subgroups for quite some time. We'll try to take also a look at the following areas as well: time permitting, immunotherapy, pan-tumor therapies, that is the tumor agnostic therapies, biomarkers, biosimilars, and perhaps beyond. And so those are the main topics that we'd like to cover. This session is going to go until about 2:55 Eastern Time, 2:55 Eastern, 1:55 Central. And we'll go to questions and answers at about 2:20 Eastern, thereabouts. So that's the outline of this session. And our first panelist is someone whom you've already met, and that's Bob Carlson, so he need not reintroduce himself. But I want to ask each of our panelists, and I'll call them out to just remind us of their name and affiliation, and what is your role in NCCN guidelines. So let's start out with Dr. Chung. Sir?
Hi, good afternoon. I'm Daniel Chung. I'm actually a gastroenterologist, and I serve as a medical co-director of the Center for Cancer Risk Assessment at Massachusetts General Hospital. And I'm a member of the NCCN panel on genetic familial high-risk assessment for colorectal cancer.
Thanks, Dr. Chung. Dr. O'Regan.
Yeah, hi, Ruth O'Regan. I'm the Division Head of Hematology and Oncology and Palliative Care at University of Wisconsin. I'm also Deputy Director of the Carbone Cancer Center. For NCCN, I'm Vice Chair of the Board of Directors on the Guidelines panel, and I'm also on the Breast Cancer panel.
Good. Thanks, Dr. Regan. Dr. Thompson. Dr. Thompson, I believe you're on mute, sir.
Of course, sorry about that. John Thompson here from Seattle, Fred Hutchinson Cancer Center, colleague of Mark. And I've been in the melanoma committee of NCCN since its inception and continue as a vice chair of that committee. I also am now the chair of the NCCN Committee on Management of Immunotherapy-Related Toxicities.
Great, thanks very much, Dr. Thompson. And Dr. Vinook.
Hi, I'm Alan Vinook. I'm a medical oncologist at the University of California, San Francisco. I'm on the board of directors of NCCN, and as with a few of our other panel members, have served on guideline panels since their inception. I'm the vice chair of the colon, rectal, anal, and small bowel, and it's still growing panel, as well as a member of the hepatobiliary panel.
Great, thanks very much, Dr. Vinook. As I said in my opening remarks, these guidelines matter to a whole range of decision makers. And I was about to say, let's kind of get into the black box here, but the box is not exactly opaque. It is somewhat transparent. And I want to ask Dr. Carlson, Bob, if you could just give us an idea about why the guidelines program in the first place, a little bit of an overview, how many panels there are, and so forth. Bob?
Sure, be very happy to do that. So the guidelines program actually started back in 1995, and it started as a counterbalance to the managed care HMO capitated patient systems that were being developed at that time. And the NCCN centers wanted to assure that they had access to patients for care. And so they told the insurance carriers, if you contract with us, we will tell you how we will manage our patients. And that's really how it got started. So currently we have 59 different panels. Each of the panels has at least 30 people on them. Some of them have 35 to upwards to almost 40 individuals. Those 59 panels are responsible for 76 different guidelines, and that number is increasing on a steady basis. We add guidelines based upon healthcare professional need and the availability of effective therapies. So a guideline is of value only if it tells you something you can do that will actually help a patient. And so availability of effective therapies, and that's why this constituency, you all that are listening, are so important to us. We need new therapies so we can develop and continue to improve our guideline experience. Each of the guidelines is updated at least annually. Because our guidelines are used for coverage determinations by the vast majority of payers in the US, if there's a new FDA drug approval, if there's a new practice-changing clinical trial reported, we do interim updates between the annual update. And so some of our guidelines may undergo 6-7 updates per year, depending upon how active the scientific findings are in that specific disease. But transparency is incredibly important to the NCCN, and there are a number of things that we do to help ensure transparency. The first is we tell you who our panel members are. Not all guideline organizations or pathway groups do. We tell you who they are. We tell you what their specialty is, what their institution is, and we disclose to you their financial conflicts of interest, if any. And I'll come back to that in just a second. But we also provide a manuscript that accompanies the guidelines that provides the rationale that the guideline panel used in making its recommendation to include something in the guideline. And then we have a system of ways of looking at evidence that helps to ensure transparency as well. Each of the recommendations on the guideline is associated with a category of evidence and consensus that tells you how good the evidence is and how uniform the consensus is among the panel that the recommendation is appropriate. We have a system called the evidence blocks where the panel scores the efficacy, the safety, the quality and extent of the evidence supporting the recommendation, the consistency of that evidence, and an estimate of the affordability of the care. And then we also more recently have what we call a category of preference that also not only if there's a pick list of 10 or 12 or 20 different options in a specific situation, the panel is asked to actually pick 1 or 2 or 3 of the options that they actually believe are the best of that long list of options. And that's the only place where the panels are actually allowed to consider cost of care in making a recommendation. If there are 2 equivalent therapies and one is very expensive and one is not, they can, because of cost, they can put the less costly therapy as a preferred regimen over the other. I told you that we worry a lot about financial conflicts of interest, and we really do. And we have a firewall, and the firewall is actually a series of concentric walls. The first part of that is that 100% of any activity that can influence the content of a guideline is funded by our member institution dues. None of those funds come from industry, none of those funds come from advocacy organizations or the government. They all come from our member institution dues. Each panel member is required on at least every 6 monthly basis to formally report their financial conflicts of interest. And those reports are published publicly on our website by individual. So you can go and see who on our panel has financial conflicts of interest and what they are. There are thresholds of financial conflict of interest that just out and out disqualify a panel member from participating on the panel. And those thresholds include honoraria, consulting, stock ownership, employment, patent royalties, any funds that would come from a device or a pharmaceutical company. Those thresholds also include the financial conflicts of interest of the panelist's spouse, their domestic partner, and their dependent children. So if a panel member is single and marries somebody in the pharmaceutical industry, they're actually bounced off our panel because they got married, because of the employment that is almost always over our threshold, which is $20,000 per company. We also have an internal firewall where people in our business development group, for instance, are not allowed to talk with the guideline staff to lobby for a change or to convey a request from a company for a change in a guideline. That's strictly forbidden among our staff. Now, we do seek input from industry and from anyone who wishes to make input to our guidelines to criticize them. And that's by our external submission process. And that's a system that is available on our website where anyone 24/7, 365 can go in and make a request of a guideline panel for a change in a guideline. Those requests are then formally reviewed by each of those is formally reviewed by the appropriate panel. And if it relates to a systemic therapy, there's also a transparency document that goes with it that actually outlines the review of the panel of the request and whether it was approved or not approved, what the vote was, and what the rationale for accepting or rejecting the request was. Those external submissions happen all the time. We have had, as of today, we have had 327 external submissions so far in 2020. So we will be well over 600 by the end of the year in terms of external submissions. So it's a system that is used quite heavily. That's sort of a guidelines 101 in
That's great.
5 minutes maybe. So
Thanks, Bob.
it gives a little bit of background.
That is good background. I just want to play off a couple of things that you mentioned, Bob, and that has to do with currency of guidelines and timeliness. And if I could go to John, Dr. John Thompson, your guideline panel is probably one of the ones that gets the most new stuff coming across the transom, John. And can you say a little bit about how confident you are that you and your panel can remain apprised of the latest and can turn around a guideline in a prompt way that will enable patients, clinicians, and even payers to make well-informed decisions? How do you stay up to date, John?
Cliff, thank you for that question. I assume you're talking about the immunotherapy toxicity panel.
Yep.
Yeah. Well, of course, the immune checkpoint drugs ascended to the world stage about 8 to 10 years ago and have changed medical oncology dramatically. And because of the activity of these drugs and the beneficial response rates in many diseases, including melanoma and others, rapidly took over as the primary modality for many cancers. Now, we didn't really have an opportunity to do randomized controlled trials looking at toxicity and interventions that would affect that toxicity. So a lot of what we do is share our expert opinions from different sites. We do our best to scour the literature, and before each annual meeting, there is a formal literature review that we all review. And then based on that, plus our personal experience, we cobble together what we think is a reasonable approach to the diagnosis, grading, and treatment of these toxicities. But we are the first to admit that a lot of this is based on that and not on prospective randomized phase 3 trials.
Mm. Well, that's an important point. And if I could bounce it over to Alan Vanook. Alan, again, the matter of the kinds of data that come in, including, as Dr. Thompson mentioned, sometimes small sample sizes, how does your group kind of, I guess it's a two-part question. One, are you able to kind of keep up with the changes in the field? And two, are you looking at evidence a little bit differently given small sample sizes and rare cancers and so forth? Alan?
Well, of course, it depends on the disease, how unusual it is. Of course, diseases are becoming, we're looking at subsets of subsets, looking at different molecular groups of patients within these diseases. So it does depend on the circumstances of the disease, where the status of the treatment might be at that time. The best example might be these agnostic marker-driven therapies that we have to deal with, or we deal with across all of the diseases. There may be just a few patients with a given disease, let's say, who are candidates for a drug, and we have to consider the data such as it is and really make the judgment based on, in many cases, consensus or best guess. It's just the way it is. Obviously, unusual diseases merit unusual treatment sometimes. Other diseases where there's obviously big randomized phase 3 trials, that's more self-evident and much easier to deal with often. Much of the challenge, though, is in everything else, which is not driven by that kind of powerful data.
So, Alan, is it fair to say then sometimes your methodological criteria or threshold have to float up or down or get modified depending upon the availability of data?
Well, certainly that's the case. Obviously, there's occasional drugs that are approved, let's say by the FDA, based on surrogate or intermediate endpoints, and you then have to decide when is it appropriate to use them and where is it appropriate. I think, again, as I said, as the fields evolve, we're making decisions, like it or not, on smaller subsets of patients, because if colon cancer, if we subdivide that into 5 or 6 different entities based on RAS status or BRAF status or HER2 status, then necessarily the decisions we'll make will be on much smaller data sets.
Got it.
And again, the truth is, as oncologists know, not a lot of care is driven specifically by phase 3 data, because in fact, the patient in front of you may often not be a patient who would have been included in the trial, and often there's not been a trial for that patient in front of you.
Got it. Dr. O'Regan, any further comments on those issues, followed by Dr. Chung, if any?
Yeah, I mean, you had asked, how do we keep up to date? Well, basically, after the major national meetings or new publications that come out, what we'll typically do is have phone calls of the breast guidelines committee, independent of the annual meeting. And we'll discuss, somebody will present the data and then we'll discuss it. And a lot of times we'll put them into the guidelines, but not always. So I really do think we keep very much up to date, because it's so important for patients and payers to have the guidelines as up to date as we possibly can. So I think we do a pretty good job of that, actually.
Thanks, Dr. O'Regan. Dr. Chung, further to that.
Yeah, the only thing I would add, I echo all those comments, is that the review process is one that's always quite continuous. So there is the annual meeting, but I do think that there's many opportunities where we review new data that comes out in between and have ad hoc meetings, so that there are times we will change guidelines throughout the year that isn't on an annual basis, when important new evidence comes out that we think is worthy of making a change prior to that annual update.
You know, this is an interesting sort of benchmark here, because in sort of the broader, maybe non-oncology guidelines world, one of the sort of formal and sometimes informal standards is update a guideline at least every 5 years. And I can't think of many of you that could wait 5 years to update a guideline, given what's going on in oncology. So in the guideline sphere, you're on a quicker cadence than others by necessity, it would seem. So that's a good take-home point, I think. I wanted to ask, back to the pandemic, Dr. Stewart did such a great job laying out the big picture there. Specific to the guidelines, has COVID had any impact on what the content is of the guidelines, how you update them, how you prioritize patient subgroups, if any, and if it has any impact, please let us know what that is. Who's the first taker on that one? I go to Ruth,
comment on that.
Dr. Thompson, and then Dr. O'Regan. Yes.
Oh, sure. Thanks, Cliff. I'll talk from the perspective of the melanoma committee, because we, of course, are using primarily immune checkpoint drugs. And the question there is, we don't know whether patients treated with those agents, pembrolizumab, nivolumab, ipilimumab, et cetera, have a higher risk of fatality from COVID or not. But certainly there's the concern about patients having to drive into the center to get their infusion. And although we like to think that our centers are safe, that is, patients are screened within the building, there's a concern about acquiring the virus through the process of this treatment. And we early on were looking at the option of different dosing regimens of anti-PD-1. There were data coming out of Merck that would allow their PD-1 to be given instead of every 3 weeks, to be given every 6 weeks. And we worked with them to come up with guidelines that would suggest that this is an opportunity to treat the patient with an effective regimen that had been tested and that would basically reduce by half the number of visits required for the patient. And so I think that's an area where we made a beneficial effect on our melanoma patients across the country.
So you did have to do a response on that, and that's a good concrete example.
Yes, and it was I think largely led by our chair, Susan Sweater, and others within the melanoma group, hoping to reduce the impact of travel and exposure for our patients.
Got it. Thank you. Dr. O'Regan, any further examples?
Yeah, I don't think we changed the breast cancer guidelines per se, but maybe we thought a little bit more about how we would apply them. So I think in the breast cancer world, things that maybe we did that are kind of part of the guidelines that we wouldn't have done before was, for example, using the 21 gene recurrence score in a kind of preoperative manner to kind of decide whether a patient would be a candidate for preoperative chemotherapy versus going straight to surgery. I think other things that came out in the breast cancer world was, for these smaller estrogen receptor positive breast cancers, using a neoadjuvant endocrine approach. So all of these things are in the guidelines, but I think maybe we're using some guidelines slightly differently or more than we would have before. And I think that would be the major change that we would see. Also, along with what Dr. Stewart was showing at the table, I think the second thing down where you have people getting perhaps, bone modifying agents and things like that, we either stop giving them or postpone them during the COVID era. So I think they would be the kind of things. So it's more not changing the guidelines, but maybe applying them slightly differently than we did previously.
Thank you, Dr. O'Regan. Alan and then Daniel on this. And by the way, I'll just kick in another kind of sub question here, and that is, if changes have been made, are there any concerns whatsoever that they could have some deleterious impact on patient outcomes? Alan?
Right. Well, that's a great question. So we actually convened sort of a subgroup of our panel and came up with a document that listed what we thought were compromises that we could live with in terms of managing patients. For example, with rectal cancer, patients need multimodality therapy. You need to somehow intertwine chemotherapy, radiation, and surgery, often in one order or another. Cancer surgery is suddenly elective. And so we had to plan well in advance for who could have a rectal cancer operation, a total mesorectal excision. Could we do radiation over 5 days, short course radiation instead of 6 weeks, minimizing the number of contacts that the patients had with the facilities and the providers? All of these factors came in. Yes, in fact, we are concerned that we may have compromised care in some way or another. It's a balancing act. You try to protect your patients from exposure. You try to protect your machines from exposure to patients who might be sick. And of course, much of this driven by for way too long inadequate testing capacity, and even today, still I think less testing capacity than we need. Nonetheless, I think we make good judgments. We're very careful and thoughtful. And even now, as we try to go back to sort of normal, whatever that is, or the new normal, we still have to think maybe there's some lessons we learned that we should put into play for ongoing care. But this is a challenge for everybody. It's a matter of, again, minimizing exposure to patients, minimizing the risk, but trying to get the best result. And hopefully we were able to strike a balance.
That's such a good point you made, among others, a moment ago, insofar as not just striking the right balance, but the COVID pandemic itself is kind of influencing or forcing some changes, some of which could outlive the pandemic itself. So you might have some procedural changes. It's interesting how a crisis sort of can have sort of permanent effects. Dr. Chung, any point to add?
Sure. Yeah, so the one comment I would make, so our panel focuses primarily on indications for genetic testing. And so for us, our guidelines per se have not changed, but I think, as Ruth had mentioned, the way we apply the guidelines have. And I think the one big thing for us has been to get away from in-person counseling and blood draws for genetic testing, and to do a lot of telehealth, remote genetic counseling and testing, and having patients do saliva kits through the mail instead of having to come in for a blood draw. So all of the same things that we do in terms of who should have testing and what the testing means still applies, but the way that we do it, I mean, these are all part of the guidelines already, but the way we do it is shifted in the pandemic era. And I think that's something that may become a more prominent part of our guidelines going forward in the future.
Thank you.
Cliff,
Sure, Bob.
Cliff, I would also add that the documents that take the parent guideline and sort of prioritize the treatments within them and strategies within them are actually posted on the NCCN website, nccn.org/COVID-19. And you'll see all the documents that the different panels have come up with to prioritize therapy, as well as other documents from the Best Practices Committee and so forth. And it's the traffic there has been pretty heavy. We've had over 17,000 users access that website since the first guideline went up. I think the second or third week in March.
Wow. So how you conduct patient visits, the role of telehealth, testing that you don't have to come in to take for a blood draw. I mean, all these could be permanent or semi-permanent changes.
Correct.
It's interesting. I had told all of our participants that we'd start taking Q&A at about 2:20, but we're getting bombarded with so many good questions. I'm going to start interspersing our discussion here with some of them. And this is going to be directed to Bob, but I really want other input after Bob kind of lays the stage here. And this has to do with, to what extent, if any, does NCCN collaborate or reflect on guidelines of other groups, such as USPSTF and other groups, ASCO and so forth? Any cooperation, collaboration, looking over each other's shoulder, Bob?
Oh, absolutely. First of all, Dr. Thompson actually was the chair of a joint ASCO-NCCN guideline panel when the immunotherapy toxicity guidelines got generated, both ASCO's and NCCN. We got together with people from ASCO and said, there's no reason why we shouldn't just do this together. And so, there's been some actually very strong collaboration. I also know that probably all the panels look at the other guidelines that are available to see if, when the new versions come out, to see if there are things in those versions that should be considered or incorporated into ours and that sort of thing. Although I will tell you that usually the paradigm is actually the other way around, because our guidelines are updated so frequently. It's usually our sister organizations or brother organizations' guidelines that are actually out of date, and they sort of leapfrog to catch up with us rather than the other way around. But we do work with others. I've been at meetings with the U.S. Preventative Services Task Force representatives recently, looking at their screening guidelines and ways of doing multi-cancer screening through liquid biopsies and so forth. So those collaborations happen all the time.
Good, thanks. John, did you want to add anything to the collaborations?
Well, I think this what Bob said is true. We also keep abreast of other guidelines, ASCO, SITC, and European guidelines that were coming out at about the same time as our early effort. In addition to what Bob said, one great advantage to our guidelines are their ready accessibility, because clinicians are in a hurry. They're in clinic, they're seeing patients in front of them, and they need to have guidance on how to approach a given situation. And I think that the NCCN guidelines are very well adapted to that, to ready access.
Good. John, actually just got a question that I think goes to you first, and this is the right timing for it since we're talking about COVID and making adaptations.
Sure.
How has COVID affected the management of immune-related adverse events? How have you been thinking about managing that?
Well, it certainly confuses the evaluation of new symptoms. One of the more feared complications of immune checkpoint therapy is pneumonitis. And so when patients present who are on immune checkpoint therapy and may already have underlying comorbidities, as Dr. Stewart was explaining in his lecture, they are not only at risk of an autoimmune toxicity, but also of COVID. And so we've had to now and throughout the different panels, throughout the different parts of our immune toxicity guideline, factor in COVID testing. And so, as Alan was just saying, we need more kits. We need the ability to do this on a real-time basis, but we have to factor that into the logic tree of how we handle a patient who's presenting with new symptoms.
Right, got it. So even those schematics, those algorithms that you show online have to be tweaked to accommodate that.
Yes.
Point well taken. Thank you. Want to move to real-world data and real-world evidence. And Ruth O'Regan, if you don't mind, want to start with you. Certainly throughout healthcare, real-world data that is consolidated into real-world evidence has taken really a front seat here. We're hearing things like use of real-world data to design clinical trials up front, to do post-marketing phase 4 surveillance and so forth, post-marketing studies and so forth, even real-world data to sometimes complement or even substitute for some clinical trials. But really curious about how your panel thinks about and uses real-world data and what the sources are. Do you use claims data or observational studies or what? How has real-world data kind of entered your thinking for your panel, Dr. O'Regan?
Yeah, I actually think this is something that's actually done very well at the NCCN guidelines panels. So basically, what happens on an annual basis is that every member institution, they're reached out to everybody in their, for example, everybody in their breast group, their surgeons, their radiation oncologists, pathologists, everybody. And they're actually asked, do they have any comments on the current guidelines? And I think that's a place where real world evidence would come in. So what would happen then is that the individual would actually submit their question through whoever their representative at the member institution is, and then all of them get addressed at the annual meeting. So certainly the breast panel it's a 2-day meeting. It's quite lengthy, but we go through every single one of those, and an individual panel member will actually take on this particular question or comment, and then it will be addressed by the whole group basically. So it's a really very thorough the way it's done. I think the way that would play in, for example, is, if we're learning something real world that we didn't expect about a particular agent that comes up, then, it's very likely that will be brought up at the panel meeting and discussed, and the guidelines will actually be changed according to that. So just giving an example from the breast panel, there's some adjuvant breast or chemotherapy regimens that are used very widely throughout the world, but maybe we don't use them very much in the US, for example. So they may be left in because they're used in other parts of the world, but they may not, depending on what the usage is. So that's kind of the way the process works. And I think it's highly effective because, particularly if you find that there's perhaps a really unexpected toxicity that wasn't seen in a clinical trial, but is seen in real world evidence, that's something that we would discuss probably at length at the annual panel. Not usually in between the annual meetings, but that's because it's done so thoroughly at the annual meeting. So I do think real world evidence is really included very well in the guidelines.
So Ruth, how accepting are you as a leader on the panel of submissions from manufacturers, for instance, who provide evidence based on clinical trials and then say, by the way, we've got some RWD for you here?
Yeah, so that happens a lot, as you can imagine. Yeah. So pretty much even sometimes in between the annual meeting as well, we'll get, information from pharmaceutical companies basically saying, this is some new data that we have, and can we include in the guidelines? And it goes through the same process. One of us will actually present that at the meeting, and then everybody weighs in and decides whether this is something that should be included in the guidelines or not, and also what level of evidence it is. So all of that gets incorporated into the guidelines.
Good, thank you. Dr. Chung, in your area, do you see any kind of viable role for real-world data?
Oh, there we go. Sorry. Yes, we do, and we evaluate that sort of on a regular basis. I do think that in our sphere, the data that we are looking at comes in the form of new genes that are identified. That there's a new gene now associated with colon cancer risk or something like that. And I think the challenge that we have mostly is that all of the very common risk genes have been already identified. So we're looking at genes that are actually quite rare. And so the challenge is that it's hard to make broad-based decisions with a lot of, based on high quality data, because there generally isn't a whole lot of data. And so, being able to take advantage of some of the experience and information that is out there does actually help. I think it requires a lot of inferences and extrapolations that the whole committee goes through to figure out how to integrate that into our guidelines. But absolutely, all of that kind of information is, I think, even particularly important when we don't have a lot of high quality data available.
So, Dr. Chung, would you be looking to look into registries of genetic information, biopsy samples, and so forth? I mean, your angle here on real-world data is a little bit different than what I've typically heard. This is great.
Right. Well, so yeah. So for us, I mean, we are not. Yeah. So registries are incredibly useful resources. I think we have actually. I think a lot of studies have actually, I will say, partnered with a number of genetic testing companies, because so genetic testing is done not sort of universally in everyone's own institutional lab, but actually really has often gets funneled to a few large laboratories. And so I think many of these companies have actually had wonderful, productive collaborations with academic institutions in order to analyze all the data that actually comes through, the thousands and thousands of tests that these labs will be doing, and as a way of really kind of learning as much as we can from relatively rare conditions.
So these massive, massive data sets from more powerful sequencing tools and so forth are just kind of give you great big piles of data, and they are real world data for much extent. So we've got to figure out how to use that. I'm wondering, Alan and John, if you have anything to add about the use of RWD, RWE. Alan?
The thing that occurs to me, Cliff, is
John.
the institutional review that the guidelines go through every year. I can't remember from Bob's presentation how many institutions there are now, perhaps in the 20s. And this, although it is a subset of the overall US cancer scene, it's a substantial sample. So it gives a good estimate of what's going on across the country. And as part of the institutional review, the entire guideline is submitted in each institution to the relevant specialists, medical oncologists, radiation oncologists, surgeons, et cetera. So we're gathering detailed input from our colleagues and then putting that together and then submitting that to the guidelines group for their annual meeting.
Got it. Point well taken. Thanks. Alan?
Well, I agree with all that. There's also the fact that the panelists, of course, bring real world experience to the table. So as you come up with the consensus decisions, it's not in a vacuum. All of these members see patients and try to incorporate that information. I think an example of where you have nothing but real world data, let's say, is these recent evidence that young people are at greater risk for colorectal cancer than in the past. So the idea of identifying younger patients who are at risk and figuring out how to deal with the ramifications of colon or rectal cancer, let's say, in people below the age of 50, a growing population of patients. Well, there's really no real rigorous data yet on that. So this is by necessity a matter of milking the data that we have, at looking at real world evidence from around the country and trying to come up with consensus guidelines in anticipation of a problem that is just now going to be formally studied to a great extent.
So that's a very helpful example of kind of a sort of an epidemiological application of real world data. Like things are cropping up as risk factors that we didn't know about before, and then maybe even case finding later on. So those data can be helpful in so many ways. Great point. Speaking of tough things to handle in the current environment, I want to move over to disparities. When we think about disparities, there are disparities in health care and disparities in health care outcomes and health outcomes and health status. And if we could kind of go to Ruth first on this one, Ruth, given the greater emphasis on understanding and accommodating and doing something about healthcare disparities and health outcomes, how does that enter at all into your panel's thinking and deliberations?
Yeah, I think the first thing I would say about this is that when you think about the now 30 NCCN sites, they really include a very, very diverse population across this country. So each of the panel members is going to bring, data from the individual patient populations they take care of. So obviously, if there is a disparity that's noted, that might be something that would be discussed at the meeting and potentially put into guidelines. Also, I think, we know that adherence to guidelines is a quality metric. And obviously, we can use that to determine if diverse patient populations receive the same care across the spectrum. There's actually a couple of fairly recent examples of that are actually pretty interesting. There was 2 abstracts from ASCO this year from essentially the same group, who basically, in one of the abstracts, they looked at the 7 of the most common cancers that are out there. And they were basically able to show that Black patients and patients from low socioeconomic status were less likely to receive NCCN guideline-directed care, and that resulted in a worse outcome. And then the same group looked at a very large number of patients with triple-negative breast cancer in California and noted that only 1/3 of them received guideline-concordant care, and that again, Blacks and Hispanics and patients from lower socioeconomic groups were less likely to receive guideline concordant care, and that this did impact outcomes. So I think they're all the kind of things that the guidelines can be used to kind of help with disparities, because of course, if we find that, these patient populations are not getting guideline directed care, then obviously trying to make sure that the guidelines are used in these patient populations would be very important. And the result of that one assume would be that you could improve outcome for those patients. So I think all of those things are discussed at the different panels, and I think they're obviously very important use of the guidelines.
So glad you set it up that way, Ruth, because you could have the most wonderful, pristine guideline, but if there's a lack of adherence, a lack of access, then what good is it? And so it's good to hear that you're kind of thinking beyond that. That raises a related point. And one of our questions has come in along these lines, and that is, you've mentioned across the 20 plus member institutions of NCCN and kind of good representation, how do your guidelines seek to account for disparities in cancer centers? In other words, community oncology centers as opposed to large academic-based center and so forth. Are the guidelines viable and applicable across these different kinds of cancer delivery centers and their resources and so forth? How can you play that? Ruth, just back to you quickly, since you were starting to get into that.
Yeah, I mean, I think the guidelines really are, at least the breast guidelines, are really specifically written in a way that they can be implemented really in most, both academic and community practices. And, of course, the community practitioners probably use them perhaps even more than we do to some extent, because we're lucky to be all disease specific in our areas. But I think that's really taken into account that as you write the guidelines, they really have to be applicable to across all the different groups of patients that are seen throughout the country. And as I'm sure Bob could talk about as well, obviously even gone further, because now we've got these global guidelines that actually have been revised specifically for patients in Africa and those type of areas as well. So I do think the guidelines, they're not put in such a way that you could own the resources are only available in academic centers. That might be the case in, for some perhaps imaging techniques, for example, but then they're unlikely to be in the guidelines if they're not widely available. So I think overall, these guidelines very much are written for anybody who treats cancer patients throughout the country.
Bob, are you confident about that? Someone might say, well, these guidelines are written for NCCN member centers, and they're not exactly a cross-section of cancer care. Would you concur with Dr. O'Regan?
Of course, I would agree with Ruth. So the guidelines originally were written specifically for the NCCN member institutions. But over the years, they really have become more and more inclusive of multiple different therapies, so that they actually should be usable by the vast majority of oncology practices, oncology centers in the United States. And if there's an area in the United States where those sorts of therapies can't be reasonably applied, then that patient probably would best be served by getting them someplace where they can be applied. Now, as Ruth suggested, we do realize that there are many places in the world, and including some places in the United States. I mean, the Navajo Indian Reservation would be a great example in the US, or Harlem in New York, where the resources are just not available in general to give guideline concordant therapy. And in those circumstances, using some of our resource stratified guidelines would be reasonable to do, although that's not their intent. The resource stratified guidelines are really designed for use outside the United States in large regions like sub-Saharan Africa, many parts of Latin America and so forth, where the resource environment is severely restricted, and applying our guidelines is just not possible. But I think that what, the message that a guideline can deliver is aspirational. This is what you should strive to do so that the system continually improves. And as Ruth outlined earlier, there's an enormous amount of literature that's available now that demonstrates that if you deliver guideline concordant care, patients do better, and counterintuitively, it actually costs less to deliver that care.
Point well taken. And thanks for raising the resource stratified guidelines. It's a good point on that. Let me, based on a question, push you a little bit harder on this one. For obvious reasons, we've heard so much lately about why African Americans are subject to these very large gaps in health and healthcare disparities. We've heard also a lot about the American Indian Alaska Native populations, including the Navajo Nation, Bob, that you mentioned. And looking at some of the data also, Latinx or Hispanic populations are hit pretty hard. Do any of your guideline panels ever say to yourselves, we need in this particular form of cancer to take a special look at any one or more of those socioeconomic, racial, ethnic groups, subgroups, and try to kind of parse the data along those lines? Do you ever sort of proactively think that way? Who wants to take that one on? Ruth, we'll pick on you. Breast cancer is so prevalent and so forth. Does your group ever say at some point, we need to think carefully about this particular patient group or that particular patient group? We kind of just start with everybody in the same bucket of data.
Yeah, it's a good question. I don't know that we've ever specifically written a guideline specifically for a patient population, but I think we certainly take into account, for example, things like, African Americans are more likely to get triple negative breast cancer at a younger age. That might lead to, in terms of what agents you use and how they're tolerated, those type of things. But I don't think we specifically have done that. I guess one of the other thing, and Daniel can probably talk to this as well, is, the whole idea of genetics, and do you think differently for patient population based on genetic testing, or groups that perhaps might be at higher risk of having a genetic mutation? Is that something that you would put into the guidelines? So I'd say in the breast cancer guys, we haven't specifically done that, but I think that doesn't mean we don't discuss it. I think just maybe hasn't quite reached the level of evidence where we put it into the guidelines yet, but I could certainly see that happening in the future.
Well, the triple negative is actually a pretty good example of kind of looking at the data available for that. Dr. Chung, what would you want to add on that?
Yeah, so this is a great point. And, I think that So as far as disparities within what we do in our group, I think one of the things we struggle with the most is access to genetic testing. And so there is definitely less access in, inner city areas, in a lot of rural areas, and much of that reflects, I think, a couple of things, but some of it is just access to genetics resources. And so, we try very hard to obviously make a guideline that we think is applicable to everyone, to the population as a whole. There certainly are going to be some genetic risk differences based on certain ethnic groups. However, I think that we have never really parsed that out specifically, because I think the level of that risk is not so high that we would, make one recommendation specifically for one ethnic group or one particular group over another. Because in general, I think many of these genetic risk factors span many different ethnicities and cultures. And so I don't think that it would be appropriate to really target one group or another. I would say we discuss it in the end. I do think that the guidelines are meant to be in many ways what we think is the optimal approach for everyone. And it did really be for us to attack the issue of how to provide the access to it, I think has become a little bit more, is a little challenging. The one thing that has been, I think, a real godsend in our field is that there's actually a lot of genetic testing that's now available, direct to consumer type of testing, that patients do not need to come to the hospital, do not need to meet a genetic counselor, but a number of companies have actually begun to offer high quality testing that can be done entirely without your practitioner. There's a lot of debate about the pros and cons of that, but I think it, with a high quality CLIA laboratory, at least the quality of the testing is, I think, comparable.
Well, I'm so glad you mentioned the issue of access to genetic testing and other resources. There may be some lack of equitable access around the country and across groups for that. Thank you. I'm wondering if John or Alan or Bob want to add anything else to the issue of disparities, whether your panel or across NCN.
Cliff, I'll just say that I have some comments that don't necessarily reflect disparity based on race or necessarily culture, but may have to be more reflected in educational level and social support. The immune checkpoint field, which is becoming so popular, is a high risk, high reward proposition. There are a lot of potential toxicities. And we put a lot of thought in the guidelines into describing that instead of just rote prescription of checkpoint therapy for a patient with stage 3B melanoma, we emphasize the importance of the patient education, patient being able to understand and comprehend the treatment, being able to communicate if there are side effects, and having the social support to get back to the center if necessary. And this also reflects back to what Mark said earlier about issues related to adjuvant therapy where the benefit may be less. There's a relapse-free survival benefit perhaps, but not a documented overall survival benefit. So we're getting a lot more nuanced in trying to present the benefits of our treatment vis-a-vis the individual patient and their educational and social support background.
That's such a great point. I mean, I was looking at Los Angeles for another issue not long ago, and I saw the list of languages that healthcare providers need to be able to kind of work in. And it's extraordinary how many there were. And so this is a big challenge for the oncology community. Bob, anything else? But I'm glad you mentioned it, Dr. Thompson, right on point. Bob, further on that, and then Alan?
Sure. So, NCCN is continually trying to improve the quality and utility of our guidelines. And, disparities like this in terms of racial and ethnic disparities, cultural disparities, socioeconomic disparities, sexual and gender minority group disparities are all places where NCCN could do better.
Thank you, Bob.
Cliff, I was going to example where it's disparities getting back to sort of are things just for NCCN institutions or for cancer centers. For example, there's a trend, there's evidence now that some patients with rectal cancer do not need surgery, perhaps. They can have definitive therapy without surgery. Well, the studies that have shown that, and there are a few of them now, really include rigorous screening and surveillance and follow-up on patients. It's not for the faint of heart. You can't have language barriers or many factors that would go into who can safely be included in this kind of approach, not to mention the facilities at the place who's taking care of the patient, the quality of the scans, all these factors. So at least in rectal cancer, for example, our panel, we've not gone there. We are aware of the literature, but do not include the algorithm in the NCCN guidelines as options because we don't believe these are realistically things that can be done on average in the community, and we wouldn't advertise them as such. Because we have to be careful. I think people, if we say we can do it in the guidelines, people, should you be trying this at home kind of thing. And so this is part of our responsibility as we create guidelines, is to make sure we're confident that what the results that we see in these rigorous studies, for example, that can be replicated in the community. And in many communities, independent of any other disparities, many communities cannot deliver that kind of rigorous follow-up or care for the patients.
These are such great points. I'm thinking about disparities in a few different ways now, and I really liked Dr. Chung's point about, for example, access to genetic resources. And a couple of you have already mentioned the issue about disparities across types of institutions within the United States as well as beyond, the issue of language, the issue of conveying difficult subjects. And Bob's comment was very helpful and straightforward, saying we can always continue to do better here. And it's good to hear that NCCN has its eye on that ball. Thank you for that. Since Alan was the one who mentioned pan-tumor therapies, we're going to go to Alan on this. And Alan, this is really a challenge, at least from an outside observer standpoint. I think Bob said that there are 59 guideline panels. And now you've got that all seem to be sort of oriented towards a particular tumor type site. Now we have this issue of pan-tumor therapies or tumor agnostic therapies. Alan, could you first explain to some of us that may not know what those are? And then I think at least one or two have been approved by the FDA. And then the third thing, I'm going to tally you up with stuff to do here, is, does the guideline structure work for pan-tumor therapies? Alan, big order for you.
So this is a challenge. There are a couple of therapies that have been approved that are agnostic to the site of the primary tumor, right? The guidelines, of course, developed, except for some of these toxicity guidelines, really the guidelines are developed per disease group. And the reason we stick with the disease groups is that's where the expertise is. So where do you place a new therapy, whether it's agnostic or not, to the disease site? If you're going to use it for a given patient, how do you fit it into the proper place in the spectrum of treatments? So the two examples, the best example are for MSI high cancers. Any patients with MSI high cancers would be suitably treated with a checkpoint inhibitor. That's based on studies that looked at that were agnostic to the sites of disease. Now, as it goes forward, though, we're picking off some of the diseases included in those groups and identifying the proper place for therapies. For example, we will change our current guidelines on the use of checkpoint inhibitors in patients with MSI high colon cancer based on ASCO data that shows clearly that a checkpoint inhibitor as first-line treatment in MSI high patients with metastatic disease confers what appears to be pretty substantial benefit to those patients. So truly, they need to reside, or they should reside within the disease, because you know how to place that treatment in the range of options. Having said that, though, then you've got the very rare, let's say the NTRK fusion patients, which is an agnostic biomarker-driven therapy, which applies to maybe a handful of patients with many different diseases. And so we elect to put that in our guidelines in colorectal cancer as sort of a footnote, where patients could be considered for this treatment at such and such a place in the algorithm in the choice of therapies. So the balancing question is, right now there are only a few of these circumstances. As they become more and more common, I would imagine we may need to change our approach. And I would also say that in fact, therapies are usually not going to be agnostic to the mutation that they're driven by. For example, the BRAF mutation, V600E, in melanoma, the patients have a very different outcome with therapy directed at that mutation than they do in colon cancer. It's the identical mutation, but in different tissues. And the results are different. The therapies, studies have looked at them, and the treatments in melanoma might lead to very different outcomes in patients with colon cancer. That's an example where you have to be specific to the disease because, in fact, the outcomes are not unrelated to the site of origin.
So if something is claimed to be a pan-tumor therapy and it is shown to work in 1 or 2 or 3 instances, you're still going to look at them on an individual basis because even if it works in 3 or more cancers, it doesn't mean it works as well across all 3.
Correct, and you don't, depending on the diseases, how you would usually line up the therapies and where it would fit in the continuum of treatment options. So that's resident in the expertise at the disease sites, and that's why, at least for the time being, we've kept them the purview of in each of the guidelines per disease, with them being mentioned across all the diseases.
Got it. Daniel, you must have a view on this. Dr. Chung.
Yeah, so I'm actually not quite sure I'm the right person to comment on this. So we, as part of the genetic, the panel, what we do is looking about germline risk genetic alterations and not looking for specific tumor marker, tumor genetic alterations that would indicate, these being indicators for these specific tumor treatments. So I think that I think that we focus primarily on not the somatic mutations that occur in the tumors, but really just the kind of germline risk alleles.
Got it. Okay. It may come to your desk at some point. Dr. Thompson,
Yes.
Any further comments on this, on the pan-tumors? You're on mute, sir, Dr. Thompson.
Figure this out. Sorry about that. Just to back up the comments of the earlier speakers, the explosion of these new genetically indicated subsets has made the creation of our guidelines more complicated. The flow diagram that we're all familiar with gets incredibly jumbled and is often crowded out by 20 or 30 footnotes. And so we're looking at ways to embed web links and so forth into the guidelines to almost make these 3-dimensional as opposed to the overly simple 2-dimensional guidelines that we've come to know.
Got it. Okay. Any further comments on the pan-tumor or tumor-agnostic therapies coming along? There's a lot of interest in the industry, of course, in those. Okay. Before we move on, because Bob mentioned gender-specific issues and so forth, right about then we got a question. Bob, if you don't mind, there was a 2017 survey on sexual and gender issues. I don't know if you recall that. That questioner raised that and wonder if there'd be any more attention in the guidelines to sexual and gender issues in how the guidelines are updated.
The short answer is yes. I'm not familiar with the 2017 survey. If whoever asked that question could send it to me, I would appreciate it so I can educate myself about it. There have actually been discussions ongoing now for maybe 2 months about doing a better job of being inclusive in sexual and gender minorities in our guidelines. We talk about women with breast cancer or women with cervical cancer, but a transgender, a person who was born genetically as a male and has become a transgender, has a prostate. So how do you screen for prostate cancer in someone who considers themselves a woman? So the language in our guidelines is not inclusive. And then there are special situations and circumstances where the guidelines could be more specific about dealing with specific issues. The issue of anal cancer and so on in gay men and so on is a different issue than it is perhaps in other groups. So there are those situations where we can do a better job, and we're at the current time trying to sort out how to do have standard operating procedures in terms of language inclusivity, in terms of gender in our guidelines. And there is discussion, although it has not gone before the guideline steering committee yet, about establishing a sexual and gender minority panel to deal with and discuss the special the cancer care issues in those special populations.
So you are looking at that, you are considering that, and that may apply to multiple guidelines, maybe not all, but certainly many of them.
Yes.
I would imagine, yeah. Several questions on biosimilars, and I know we actually planned on talking about this, but we have a lot of interest. And it looks like people want to, let's go to Daniel and Alan on biomarkers, and just basically among the proliferating number of biomarkers, how do guidelines assess their clinical utility? Their clinical utility. My recollection is that analytic validity is how good you are at finding a biomarker. Clinical validity is how well does that biomarker act as a predictor or prognostic. But clinical utility is how well does a biomarker inform medical decision-making and patient outcomes. And I wonder if we go to Daniel and Alan on this, thinking about clinical utility of biomarkers, how that thinking is emerging. Daniel?
Right. So, yeah, thank you. So, so in our panel, what we're looking at primarily are how do we evaluate new genes that are being identified that are associated with colon cancer risk. And so this happens on a pretty regular basis, that people will say a new gene such as MSH3, NTHL1 or something will be associated with a higher risk of colon cancer. And so our panel, just to step back, is really, I focused on trying to identify, what are the indications that someone should have testing for a certain gene? And then ultimately, how do you manage someone who has one of these gene mutations? And so, again, I think I made this point earlier, part of the challenge has been that, these mutations end up being relatively uncommon. We see things like Lynch syndrome, which is far away the most common hereditary cancer syndrome, and colon cancer syndrome, and we have a lot of data to guide us there. So for these other more rare diseases, we are really kind of, I think, left with a lot of very limited evidence that we can use in terms of defining how do we make use of this information. And in the end, is the questions that arise for us is, number one, how often are these mutations found? Can we actually quantify what the specific cancer risk is for carrying one of these mutations? And then what data are there in terms of implications for management afterwards, notably and mostly in terms of like what kind of screening and surveillance would you do, high-risk screening and surveillance would you do an individual who carries one of these genes? And so for us to include one of these genes in our recommendations, I would have to say the whole field has changed a little bit, because we no longer put out guidelines specifically about testing for one gene or another. The way the field has migrated is that we now do these so-called large gene panels, where we will say if someone wants testing for colon cancer, they will get a panel of 20, 30 colon cancer genes. And so often what happens is that each of these new genes just gets added on to that panel. For us to then interpret what you make of those particular results, I think relies very heavily on whatever small amount of evidence is available. But what we, I think, often do is that we will make a lot of inferences based on similar genes and other hereditary syndromes. I think that in the end, for us, clinical utility is going to be ultimately defined by knowing that each of these, that passing a certain threshold for increased cancer risk, and that it actually has implications for how you change your screening or surveillance afterwards. So I think that's the main piece of what we do in terms of using new genes as and considering them as biomarkers.
So you are thinking through, when new genes come before you, related biomarkers come before you, you're thinking, how are we going to make use of this information? Whether
Correct.
It's further screening or surveillance, and even down the road to treatment and outcomes. You are
Correct.
thinking that, but you also made the point that you're looking at panels of these, colorectal, whatever form of cancer,
Right.
might be 20 or 30, and sometimes new ones are kind of just added in, tossed in, and maybe you didn't have a chance to look at every single one that way. But the whole panel still works, right?
Correct. And so I think that this is where I think we've evolved to the concept of panels with so-called actionable genes, where we have a lot of data, where we know what the risks are, we know that it changes management, and the ones where there's, we would say, sort of limited data and uncertain implications. And these are obviously these panels that are now hundreds of genes on them, and it turns out that some patients want all that information, even though we don't quite know what to do with all of it. But we do try to make, as far as our guidelines go, we do try to provide some education, really, and guidance about what you can learn from each of these types of panels and what you should expect if you choose one type of panel or another.
Got it. Thanks very much, Dr. Chung. Dr. Vanook, you must have some views on this.
Well, this is one of the big challenges. In fact, I think it's harder many times at our meetings to come up with, to resolve issues around diagnostics, harder than it is to resolve the same issues around therapeutics. We come from centers where we typically do multi-gene assays. We do these 500 or 600 gene platforms that look at cancers, look at the somatic mutations in cancers. There are commercial outlets that do these all over the place. You've got blood tests to look for circulating tumor DNA. All of these factors can generate a boatload of many biomarkers and factors, but our goal is to figure out really what matters and what tests can we do that would help us make a decision. So at least in our panels, and I think most of the panels, we try to be extremely rigorous, because you can spend a lot of money and not get anywhere, and in many cases get a lot of misinformation. I think one of the biggest problems we have with biomarker-driven therapies are the therapies that are done to target a biomarker that are totally inappropriate, either unproven or proven not to work. But let's say a commercial test will come with a mutational finding, and there'll be a comment in the discussion. You might consider A, B, C, and D to treat this mutation, even if there's no evidence that treatment would work. This is the way the system is. And so we try to be very rigorous and also be aware of the reality, which is, in fact, our goal in colorectal cancer, for example, would be to have every patient tested for MSI status and for RAS status. If we could just accomplish that, we'd be happy. But in fact, even if you look across the country, still 30% of patients with colorectal cancer apparently are not getting KRAS analysis on their tumor. This is a failure of education. But so we sort of, either the extremes really are the problem. You have people not getting even the most necessary things, and many other patients, many times driven by their own wishes or by their cursory read of the literature, coming in with way more information than we need and trying to make decisions that are way more complex than they should be because of the biomarker information that doesn't help us.
But it sounds like there are some disconnects here between biomarkers and utility here. I mean, sometimes biomarkers that ought to be assessed aren't, which is not good for patients. And sometimes you've got the opposite situation where they're just tossed in, they're going to provide information that might even be misleading. I'm wondering, Ruth and then John, any further views on biomarkers along your lines in your panels?
Yeah, I mean, I think it's all about being practical here, right? So to give you a couple of examples, I mean, I think you want to make sure if the biomarker is important, it's something that's accessible. We were talking before about, making sure it's accessible to the community. So an example of that would be, PD-L1 testing in the immune cells for triple negative breast cancer. Do you just say you have to use one particular biomarker for that, or can you use different assays? So I think things like that, making it easier for patients, I think they're very important. But I think also, is this something important for patients right now? So an example of that would be ESR1 mutations in ER-positive breast cancer. We talked about that a few years ago, about putting it in our guidelines. But at the end of the day, it really wasn't going to impact how we were going to manage patients at that time point. I know we'll revisit it, but again, putting in the guidelines at that time point didn't make any particular sense, because it really wasn't going to be something that would be impactful for patients. So I think it's all about being practical and basically working out, making sure the biomarker is something that really is going to influence patient care.
So you continue to ask that question. Great, thank you.
Yes.
John, anything to add on that, on the biomarkers, John, from your standpoint, your panel?
Yeah, we struggle with this as well. And you mentioned the clinical utility of biomarkers. That's an important endpoint. It's a moving target in melanoma, for instance, because the AJCC version 8 staging system gives us a better idea of the prognosis of a patient for a given readily ascertainable clinical factors. Now, in that context, what is the value of, say, tumor gene expression profiling? Well, we have to look at that in the context of where we are now with our current staging as opposed to old staging systems.
Aha. I have a very specific question that came in on clinical utility here, and it has to do with the PD-1s, PD-L1s. How does NCCN assess the clinical utility of new ways of assessing established biomarkers, for example, imaging a particular biomarker such as PD-1, PD-L1, or an estrogen receptor that visualizes biomarker distribution heterogeneity compared to a single site biopsy or blood tests that average out biomarkers across all tumors? Pretty sophisticated question, though. Back to the PD-1s, PD-L1s. Thoughts on that?
Well, that would certainly have to be assigned to a subcommittee of a given panel, and this might happen at multiple disease panels. This is a very technically challenging question you're asking, so that we could really become fully informed and then report back from an independent perspective, from the NCCN perspective, to the respective panels,
Right.
the value of this.
This will be pretty technical. It's just really talking about visualizing biomarker distribution heterogeneity versus single site biopsy or blood tests. So there's a lot going on there at that point. Something to be brought up, but it does sound like there's a place where you could go to try to seek an answer to that question, John. Thank you. Speaking of, yes.
Our panels are multidisciplinary to the nth degree. And so we have imaging folks, we have pathology, and we either have the requisite expertise within our panels or we find it when these kinds of issues come up. So we find this is one of the most challenging things, but one of the most important things we can do, which is to put in the proper perspective new technology, rather than rushing to put it into play, making sure it's worth it and we understand it. And so we at every meeting we'll have subgroups that will talk about these kinds of nuanced technical things to put it in perspective.
But what's emerging, it seems, is that there's sort of a triaging function that goes on across the 59 guideline panels. And then again, the pan-tumors was a good example of kind of thinking, where does a pan-tumor go? And then within panels, you've got to triage to the appropriate types of expertise, whether it's imaging or something else. A point well taken, Alan. We have had multiple questions come in on biosimilars. And I think maybe starting out with Bob, because Bob, what, from an NCCN organizational standpoint, does the organization have a stance on biosimilars or a policy or any kind of single orientation on that?
Yeah, we do. So we started experiencing biosimilars several years ago, and the NCCN approach at that time was to have each individual panel look at each individual biosimilar and understand how to incorporate that or not into the guideline. As experience with biosimilars has become more widespread, and as there's an increasing number of biosimilars, people have become increasingly comfortable with them. And the Guideline Steering Committee, actually this last March, during a web conference, voted to include in the NCCN guidelines anywhere that an innovator compound is listed, if there are FDA-approved biosimilars, to add a footnote that just says, you list the innovator compound in the guideline with a footnote that says, and an FDA biosimilar is appropriate to utilize in place. So it basically, that's not the exact language, but the gist of the language is that any place an innovator compound is included, the NCCN now believes that an FDA-approved biosimilar is an appropriate substitute.
So is the term approximately an appropriate substitute rather than any anything that's mandatory or required?
Yes, it's an appropriate substitute.
An appropriate substitute. Good.
And should there be some concern about a specific disease site and a specific biosimilar, then the panel would have the authority to say, but don't use the biosimilar in this circumstance. But at least as of today, everywhere in our guidelines, we know that there's a biosimilar approved. The panels have allowed the use of the biosimilar.
Got it. Thank you. Alan and others, comments on biosimilars and how they're being absorbed into your guidelines.
Well, I think Bob characterized it correctly. That is a policy. Now, in terms of how different places apply it, right, these are just guidelines. They're not rules. And so, for example, you might look at a drug that is a biosimilar recently approved based on one study in one disease in combination with a chemotherapy, let's say, and not to be mentioned biologic biosimilar in lung cancer with chemotherapy that's been approved based on that study, based on comparable results to the native biological agent with chemotherapy in the same disease. Well, that agent may be used in many other diseases, in many different doses, different schedules with different chemotherapies. We do as a guideline panel, we acknowledge the interchangeability of these biosimilars. But remember, back home here in San Francisco at UCSF, we may have a different judgment whether we agree that this can globally be accepted. As Bob said, it's okay, it's authorized. You can do it, but you aren't mandated to do it. And I think now whether we're looking to count how many angels can dance on the head of a pin is a different question. Are these really relevant differences? We don't know. But certainly if we're not comfortable with interchanging a biosimilar, we'll make that decision at our home base. But the guidelines are inclusive because based on the data as it stands, these are felt to be appropriate therapies, and we don't know otherwise. There may be biases that tell us to worry or to think twice, but in fact, we don't know that that's correct.
Understood. Good. Thank you. Any other comments about biosimilars at this point? Okay. We've got about 5.5 minutes left, and we've gotten a few questions on kind of the payment and economic stuff. So I'm going to try to fit at least one of these in. This has to do with the relationship between categories of evidence and payment or reimbursement. And I know that none of you is a payer, but you do work in guidelines and you do set out categories. And so here's a question, please follow us on this one. We understand that category 2A and 2B recommendations are both based upon lower level evidence, with category 2A having uniform NCCN consensus greater than 85%, and category 2B having NCCN consensus greater than or equal to 50% that an intervention is appropriate. And at least one person has questioned whether you want that distinction between 2A and 2B, because they're saying we have experience that a category 2B recommendation is used by payer contractors and private insurers, CMS contractors and private insurers, as a reason to deny coverage and payment and therefore deny access to these technologies. It seems that they only recognize a 2A as something that ought to be basically covered and not as opposed to 2B. And this is arising in some areas such as genomic testing and so forth. I won't go through all of it, but from the standpoint of what you intended in the categories of evidence, do you mean to have that kind of distinction between 2A and 2B? And again, I know you're on the clinical side, but do you have any thoughts about whether something ought to be covered as a 2A versus a 2B and how that might be affecting your own medical decision-making? Any takers? We'll pick on Dr. Well, go ahead, Alan.
Oh, I mean, I can tackle this. This is a very tough one. It depends on the details and the circumstances. But obviously, if 85%, the rationale, I think it's pretty solid. If everybody at the table in a multidisciplinary group at a meeting or on a conference call agrees that the evidence supports a treatment and everybody agrees, and then I think that's a very different metric than, let's say, half the people agree and half the people disagree. Whether it's a unique to a given institution or the biases of an institution or the data is seen by different people in different ways. So our job isn't to figure out how to cater to the payers. Our job is to figure out what we think is the right thing for patients. And so we're aware of that, but I think we generally try to do what's right and what's best. And again, on average, the belief being that the sort of 2A and 2B reflects the status of the field. If everybody, if more than 50% or half the people don't agree that something should be supported or is supported, then perhaps it should be left up in the air. I mean, you hate to deny patients treatment. In some diseases, for example, where it really does matter what order you do things in, there may be others who disagree and think that your choice for second line should be first line and vice versa. You don't want to give in to that if you actually have data to support the contrary. So I think it's a real challenge, and we do understand the ramifications, but we don't spend a lot of time talking about that at our panel meetings, I must say.
Thanks, Alan. Any further comment on that?
Yeah, I would. This is Bob. So if you look at the 2A recommendations, and if you're at panel meetings, the vast majority of 2A recommendations are unanimous. It's occasional that there's one or two outliers, but usually they're unanimous. The 2B recommendations, by definition, are not. And so I think you should really view the 2A versus 2B as a rank ordering of different alternatives that are available. And I'm not aware of a single circumstance where there is only a single option available that's categorized as 2B. The panels do want therapies to be available and to be paid for. And I would also point out that there is a little bit of shifting in the field now in terms of how some payers are looking at category 2Bs. Of course, United Healthcare has always paid for category 2B recommendations. One of the subtleties in the recent Medicare administrative contractor, the Palmetto group, is that their draft guidance in terms of systemic therapy coverage in cancer now their draft guidance extends down to category 2B. So we're actually seeing a drift within one of the dominant MACs in terms of CMS coverage.
Good, thanks. Very important to know, and I think that the instance that you just mentioned, Bob, was probably on the minds of the person who posed the question. So let it not be said that we're not giving you the tough questions as we get them. I want to make this a closing question, and I'll go a couple minutes over 55 after the hour, because at least 3, maybe 4 questions have come in that are the following. And I want to ask each of you to answer it in just a sentence or two, please, not anymore. And the basic question is, I'll paraphrase the 3 or 4, is basically, okay, a lot of us in your audience today are from industry. We're glad that you've explained the guideline process to us as clearly as you have. What can industry do to be a more informative, collaborative partner of the NCCN guideline process to render more effective, efficient, and safe guidelines for patients? I just gave you a distillation of about 3 or 4 questions, but I'll start with John on this one. John, you've been doing this for a while. When you think about the role of industry, how could they be better partners or collaborators? A sentence or 2 most, please.
The industry submissions are critical. We prioritize them at every annual meeting. And so providing as much data as possible would be great.
Good, thank you very much. Ruth O'Regan, Dr. O'Regan.
Yeah, I'm just going to agree with John. I think that, supplying us the data, for the panel meetings is very important, and then we can discuss them. And I think that's really a huge service that they provide to us on the panels. So.
Thank you very much. Dr. Chung.
I would agree with those comments. The only thing I would add is just I think it's really productive to be able to have strong research relationships as well.
Good point. Thanks for that as well. And Alan?
I would agree with that, with the exception of asking industry not to provide data that doesn't support, in the minds of most anybody, the inclusion or the elevation of their treatment to the level they'd like. Give us good data and solid data, which is almost always what we receive. But there are examples of sort of, gee, if only you would do this, and that clutters up our schedule a bit, but that's the only caveat I'd say.
So you want the best evidence and you want it on target as well, Ellen. Thank you.
Yes.
And Bob, would you agree with all the above, or did you have anything to add about relationships, collaboration?
So I would agree with all of the above. You know, give us good drugs, supported by good, solid data, and they'll get on the guidelines.
Got it. Got it. Well, as I said a couple minutes ago, let it not be stated that this group has not taken the tough questions. I thought I had a bunch of challenging questions ahead of time, but we had, I'm counting about 6 to 9 questions in addition that they fielded that I wasn't quite expecting, or combinations of those. And there was a straightforward answer on every single one of those. So in a moment, I'm going to turn it over to Gary Waymuller to close, but I really want to express to our panelists and really our audience. We had several hundred people here today listening into this discussion, and it's clear that industry and other stakeholders have a vital interest in not only understanding these guidelines, but in helping to ensure that they are transparent, that they are evidence-based, and that they adapt over time to challenging conditions. And certainly the pandemic has really challenged the process. And what at least I heard is that across the board with our guideline panel leaders here with us, they have learned, they've adapted, they continue to evolve. And overall, from Dr. Carlson's standpoint as well, the organization has been able to evolve accordingly. And so with that, Alan Vinook, John Thompson, Ruth O'Regan, Daniel Chung, thank you much, Dr. Thompson as well. Thank you very much for all this great input. It's very generous of your expertise and your time to share this with us, along with Dr. Carlson as well. And with that, I want to turn it over to Gary.
Thanks, Cliff. First of all, I wanted to thank all of you attending in the audience today. It's always a pleasure to have such a great attendance and such a great crowd like this. Sorry we can't see you all in person, but virtually works just as well. I'd also like to echo Cliff's remarks of thanks to all of our panelists for their time and participation on this panel. Very informative, and it's great to have you all here. I also want to extend thanks to Mark Stewart for his presentation, and of course to Cliff Goodman, our moderator, who does a fabulous job of helping us bring all this together. So with that, I just have a couple of things I'd like to tell you about. And Mark, if you could go to the next slide. We have a few events coming up that I'd like to tell you about. If you're interested in learning more about, you heard today about the clinical guidelines, but if you're interested in learning more about how the guidelines for patients are adapted and updated from the clinical practice guidelines, we're having 2 events in support of the NCCN Foundation, July 24th and July 30th. So if you are interested, registration is open now for those events, and please sign up. Next slide. So our policy group has been very busy in terms of all the recent legislation that's been taking place. We recently had a policy summit on June 10th that was well attended. We have 2 more that are coming up. The first one is on September the 9th, and it will involve innovative solutions to drive down healthcare costs. We also have our Patient Advocacy Summit, which will be coming up on December 10th. This is probably our best attended summit and fills up very quickly. So both of these will be live webinars, and if you're interested, we'll have registration for this very shortly. Next slide. So in addition to our NCCN Academy, we also offer a global academy for our industry partners that have global interests. We have a number of global corporate council members who participate, and this is open to others. This will be a live webinar on September 16th, and registration will be opening very shortly for this as well. If you're interested, there have been a number of different topics discussed, and we're in the process right now of finalizing this agenda. And lastly, the NCCN Academy that you heard today we will be offering our next NCCN Academy in October, on October 6th. It will be a virtual program, and registration for the Academy is open as of today. So, and that's it. Again, I thank all of you for your attendance and for hanging in there with us today. Also wanted to give kudos to our business development team under Elise Desjardins and their efforts to put this program together. And lastly, our meetings team under Mark Geisler for pulling it off. So thank you all for attending, and hope to see you at the next Academy in October. Thanks.